So this is Lab 8, Carbohydrate Catabolism by Monica Maccaro and Darius DeWitt. This is the fermentation PowerPoint. So anaerobic respiration is used when oxygen levels are either too low or being used faster than they can be replenished, like when you're working out, for example. Now some molecule other than oxygen will have to be used as a final electron acceptor during the electron transport chain, because it obviously can't use oxygen because it's without oxygen. And so humans cannot undergo anaerobic respiration. We can undergo, however, muscular fermentation, which produces lactate. And yeast also undergoes fermentation, but it produces ethanol, so it's a slightly different process. So the type of fermentation that we are experimenting here is we will end up with the ethanol, because we were testing with yeast and the different sugars. So so fermentation only goes through glycolysis, and it needs a lot of NAD+. Now, to get the NAD+, it first has to reduce pyruvate using the NADH, as you can see through this process right here. So many different sugars can be used for fermentation. However, glucose is the preferred sugar. So pyruvate is reduced, and ethanol and CO2 are the products of this form of fermentation. So for the procedure, we first got 4 beakers with 15 milliliters of sugar each, and so the first beaker contained glucose, the second beaker also contained glucose, the third had fructose, and the fourth had lactose. So we then added 0.5 grams of yeast to beakers 1, 3, and 4. And after that, we put the substances that were in the beakers into the swan tubes, as you can see in the picture right here. These are the swan tubes. And so we then covered the swan tube with our thumbs, flipped it upside down, and what this did was it freed all of the locked air. When we filled it up, there was locked air in the top of the swan tube, so when we flipped it upside down, all the air was released. We then incubated all of the swan tubes for 37 degrees Celsius for an hour, and we recorded the size of the carbon dioxide at every half hour. So at the end of the experiment, this is what we got. So as you can see, test tube 1 with the glucose had a lot of carbon dioxide, as well as test tube 3. However, neither test tube 2 nor 4 had any signs of carbon dioxide. So at the half hour mark, you can see that our first test tube had about 15 milliliters. The second one had 0. The third had 10. And the fourth had none. And now the difference is at the 1 hour mark, the first one had 77. The second had 0. The third had 40, and the fourth had none as well. And so we concluded that glucose is the most preferred sugar, and it's demonstrated here because it, it because there was so much more carbon dioxide at the end than in any of the others. For the glucose without yeast, well, fermentation needs something to start that process, and without the yeast, there was no way for it to possibly have started, and therefore, of course, no carbon dioxide would have been made. Fructose is another sugar that can be used, however, is not necessarily preferred because, as you can see, glucose was a lot more effective. And lactose cannot be used for fermentation whatsoever. Thank you, and Monica will pick up with the other project.